The overall goal of the following experiment is to track the microstructure of the breast to detect breast cancer using a completely non-invasive and safe imaging method. This is achieved by taking MRI images of both breasts and measuring in the entire breast fibroglandular tissue the water diffusion tensor to reveal the normal and pathological breast microstructural and cellular features. As a second step, the diffusion tensor imaging datasets are processed using a symmetric tensor model in order to calculate the directional diffusion coefficients and the anisotropy indices pixel by pixel. Next, vector and color-coded parametric maps of the directional diffusion coefficients, their average apparent diffusion coefficient, and the fractional and maximal anisotropy indices are produced to identify the best parameters for detecting malignancy and examining pathology. The results show that the diffusion tensor parameters can reveal the breast architectural features and detect malignant growth by finding map regions with reduced diffusion coefficient values and reduced maximal anisotropy values compared to normal tissue. The new diffusion tensor imaging method showed a breast cancer diagnostic accuracy equivalent to the standard dynamic contrast enhanced 3-time point method.
The main advantage of this technique over existing MRI methods like dynamic contrast enhanced MRI is that DTI does not require injections of an external contrast agent, and therefore it is completely safe and non-invasive. It is also a relatively fast method and is clinically